Teach me some meiosis, teach me some meiosis, teach me some meiosis, teach me some meiosis.
Welcome to another virtual biology lecture. Today we're going to be discussing meiosis and mutations. Hope you guys enjoy and don't forget to take good notes. Let's first discuss chromosomes a little bit more. As we previously mentioned, each species has a unique number of chromosomes per cell. Humans, for instance, have 46 chromosomes in somatic cells. In other words, your body cells. But what does that even mean? And what about germ cells or sex cells? How many chromosomes do these have? Well, you might remember that we've mentioned homologous chromosomes in previous lectures, in which we said that each of your body cells contains a set of 46 chromosomes, which come in 23 pairs known as homologous pairs. Now remember that somatic cells are genetically identical to each other unless mutations have occurred. Suppose that you have 23 pairs of socks. You would have a total of 46 socks that you could divide into two sets, 23 right socks and 23 left socks. Similar, you have body cells that have 23 pairs of chromosomes for a total of 46 that can be divided into two sets, 23 from your mother and 23 from your father. Just as we use both socks, your body cells use both sets of chromosomes to function properly. A complete set of chromosomes is called a diploid set. So in humans, that would mean 46 chromosomes. Well, somatic or body cells have diploid set of chromosomes, and sex cells, on the other hand, they only have half the set of chromosomes, which is why we refer to these as haploid. So in humans, that would mean 23 chromosomes in their sex cells. These sex cells are haploid because eventually they're going to go through fertilization, in which the haploid set of chromosomes from the dad will unite with the haploid set of chromosomes from the mom and develop a zygote, which is basically a diploid set of chromosomes. That means that each pair consists of one chromosome from each parent. Overall, humans have 22 homologous pairs, and these are referred to as autosomes. Now, these chromosomes contain genes for characteristics that are not directly related to the sex of the organism. But then we have the 23rd pair of chromosomes. These are actually referred to as sex chromosomes, and they do have a direct control of the development of sexual characteristics. Now, in mammals, including humans, an organism with two X chromosomes is a female. An organism with one X and one Y chromosome is a male. Despite the X and the Y chromosomes pairing up with each other, they're not really homologous. The X chromosome is actually larger than the Y chromosome. The Y chromosome is actually the smallest chromosome. The formation of new organisms of the same species is called reproduction. Overall, there are two types of reproduction: asexual reproduction and sexual reproduction. Now, we've discussed asexual reproduction a bit in previous lectures. This is actually the reproduction that requires only one parent, and most single-celled organisms like bacteria use this form of reproduction. It's known as binary fission. Most eukaryotes, including humans, use mitosis as a form of developing new somatic cells, also known as body cells. Remember that all DNA is copied during interphase, and then one copy is distributed to each new daughter cell. In humans, this results in each daughter cell consisting of 46 chromosomes, which means that both the parent cell and the daughter cells are diploid. Overall, mitosis is used for development, growth, and repair of old or damaged cells. Just remember that DNA in somatic cells is not passed down to offspring. Therefore, mutations in this DNA will not be passed down to their offspring. However, if the DNA in germ cells is altered, then this mutation is actually passed down to the offspring, which leads us to the cell division involved in sexual reproduction called meiosis. This process occurs in germ cells, which results in the production of gametes. Meiosis makes genetically unique haploid cells from a diploid cell. Now, these haploid cells undergo more processing in the ovaries and testes, finally forming mature gametes. But before we get to that, let's take a closer look at the steps involved in meiosis, which are very similar to those seen in mitosis. For instance, cells must undergo interphase in both processes, whether the cell will undergo mitosis or meiosis. Thus, the first stage in both mitosis and meiosis is interphase, which is where the cell grows, DNA is replicated, and then the cell prepares for cell division. Remember that at the end of interphase, DNA has been replicated, meaning all 46 chromosomes have been duplicated. So now you have 46 chromosomes and 92 chromatids. Now, if you remember from mitosis, this cell division involves 4 major steps: prophase, metaphase, anaphase, and telophase, which are referred to as PMAT. This is also seen in meiosis, except there are 2 rounds of division known as meiosis 1 and meiosis 2. Basically, meiosis 1 consists of the cell undergoing prophase 1 with 46 chromosomes and 92 chromatids. Remember that in metaphase, chromosomes line up in the middle and spindle fibers attach to the centromeres. Well, this still happens, except that homologous pairs of chromosomes line up at the middle in structures known as tetrads, just like these seen in this image. Then the spindle fibers attach. Now, during anaphase 1, the homologous chromosome pairs are split up, resulting in 23 chromosomes with 46 chromatids on opposite sides of the cell. Then in telophase 1, 2 haploid cells are produced in which each new cell has 23 chromosomes with 46 chromatids. Then these 2 cells undergo cell division once again, known as meiosis 2, starting with prophase 2 and leading to metaphase 2. But notice that in metaphase 2, the chromosomes will line up in the middle again, but now when they separate during anaphase 2, a chromatid of each chromosome will move to opposite poles. This means that in telophase 2, 4 new sex cells, each with 23 chromosomes with 23 chromatids, will develop, in other words, haploid cells. Remember, this is done because when male and female sex cells combine to form offspring, each sex cell contributes half of the normal number of chromosomes. But how is it that sexual reproduction produces such a diverse set of individuals? Well, that's because genetic variation results from both meiosis and mutations. Recall that during meiosis 1, homologous chromosomes pair up in structures known as tetrads. Well, these structures are actually formed in prophase 1, in which one homologous chromosome that comes from the individual's mom, such as this purple one, and one from the individual's dad, such as this blue one, come together and form a homologous pair. This is actually the stage in which chromosomal variation may be seen through a process known as crossing over. Now, crossing over involves DNA segments of non-sister chromatids. In other words, one chromatid from a purple one and one from a blue one. This is where DNA is exchanged between the homologous chromosomes, like seen here. We call these new chromatids recombinant chromosomes. Now, keep in mind that crossing over can actually happen in different regions of the chromatid, giving different recombinant chromosomes. This is actually why siblings are not exact copies of each other or of their parents, because the possible outcomes of recombination are numerous. As we mentioned before, mutations may take place during DNA replication or cell division. When these mutations involve body cells, they're actually referred to as somatic mutations. Now remember, somatic mutations are not passed down from parent to offspring, and they can only affect the individual. On the other hand, a mutation that takes place in a gamete or a germ cell is sometimes referred to as germline mutation. Now, germline mutations can be passed down from parent to offspring. They can affect the survival of an organism and change the traits of the organism's future offspring. In humans, germline mutations cause disorders such as hemophilia and color blindness. Now, this happens because sometimes chromosomes experience breakage in which they may gain, lose, or even inverse genes. We call these chromosomal mutations. For instance, let's take a look at this chromosome, which has various genes labeled on it. For whatever reason, the chromosome breaks, and before it comes back together, an extra copy of gene B is added. We call this type of chromosomal mutation a duplication, because a gene was duplicated and caused the chromosome to increase in length. However, what if instead of adding an extra copy of gene B, it was actually removed? Well, this would be referred to as a deletion. And you will notice that the chromosome actually becomes shorter. Now, there are occasions in which a segment of the chromosome is actually reversed, causing it to not follow the same gene order. We call this type of chromosomal mutation an inversion. It's important to note that no genetic information is actually lost or added here. But if the break and the reversal occurs at a crucial point in that chromosome, then it can actually cause serious consequences. This type of mutation could even be lethal. Okay, lastly, we have translocation mutations, in which they should not be confused with crossing over. Crossing over happens with two homologous chromosomes, and they're exchanging information regarding the same genes. Translocation, on the other hand, involves the exchange of information, but with non-homologous chromosomes. In other words, it's involving different genes. Chromosomal mutations change the number or sequence of the genes in a chromosome. Now, it's important to note that all mutations change an organism's genetic information.
Sometimes chromosome numbers are affected during the formation of gametes due to mistakes in the separation of homologous chromosomes or of sister chromatids during meiosis. Now, we call this mistake a nondisjunction, meaning the chromosomes did not separate normally as they should during meiosis, as seen in this image. And as you can see, there are two types of nondisjunction based on what failed to separate. The first is a nondisjunction of homologous chromosomes during meiosis. Basically, the homologous chromosomes don't separate for whatever reason, resulting in one of the cells having an extra chromosome and the other having one less at the end of meiosis 1. Then, once meiosis 2 is complete, all gametes produced will either have extra chromosomes or be missing chromosomes. Now, the second type of nondisjunction occurs when sister chromatids don't separate properly during meiosis 2. In other words, meiosis 1 produces two normal cells, which then go through meiosis 2, but this is where the mistake happens, in that one of those cells in which the sister chromatids don't separate, resulting in two cells with incorrect number of chromosomes. One will have an extra and the other will be missing a chromosome. Just keep in mind that the other normal cell will produce two normal cells out of it. Now, this is why this type of nondisjunction does not affect all of the gametes that are produced. Geneticists use karyotyping techniques to determine if a nondisjunction has occurred in an individual. For instance, as we said before, humans have 22 pairs of autosomes, and the 23rd pair of chromosomes is the sex chromosomes. And as illustrated in this picture, this individual is a male because he has an X and Y chromosome. So what do we call this picture of all of those chromosomes that are paired up with their respective homologous chromosomes and then arranged by size? Well, we refer to this as a karyotype. And geneticists look at these karyotypes of individuals so that they can determine if the individual has any abnormalities in the number of chromosomes that are present. For instance, this is an example of an abnormal karyotype of a male with an extra 21st chromosome, which is an indicator of Down syndrome, or such as these examples in which the number of sex chromosomes is affected, resulting in certain chromosomal disorders.
Usually, the process of DNA replication and meiosis happen without mistakes. However, mistakes do happen. Those mistakes are called mutations, which may occur for many reasons. Some mutations result from spontaneous errors during DNA replication or cell division. However, there are also other factors to consider, such as environmental conditions. For instance, radiation, tobacco smoke, and other pollutants can change cells and disrupt the cell cycle, which may lead to mutations. Now, we tend to call these agents mutagens. Some cancers, such as skin and lung cancer, are caused by mutagens such as the sun's ultraviolet radiation, or chemicals in tobacco, or asbestos. Overall, mutations can increase genetic variation and provide genetic diversity within its species. Some mutations are neutral, meaning they lead to traits that neither help nor harm the organism. On the other hand, there are other mutations that can harm or even kill the organism that inherits them. Still, other mutations are considered beneficial. For instance, some germline mutations may provide their descendants a beneficial mutation which may help an organism survive in a changing environment. Now, these mutations are likely to increase in frequency and persist in the population. Evidence has actually showed that the beneficial mutations actually contribute to evolution and speciation. Okay, now that we're familiar with what mutations are and the two types of cell divisions, let's take a quick look at what our major differences are between mitosis and meiosis. Now remember, mitosis is the cell division involved in producing somatic diploid cells from other somatic diploid cells. In humans, this means that the somatic cell begins with 46 chromosomes and ends with two new identical somatic cells, each with 46 chromosomes. Now, this is possible because DNA replication takes place during interphase, which gives a total of 92 chromatids, 2 per chromosome, allowing the chromatids to split during mitosis. Okay, what about meiosis? Well, this cell division is responsible for producing 4 unique haploid sex cells from a diploid germ cell. In humans, germ cells must also go through interphase in order to begin this cell division, with 46 chromosomes and 92 chromatids. Meiosis undergoes two cell divisions in which prophase 1 pairs up homologous structures in tetrads in order to allow crossing over to occur. Then following metaphase 1 and anaphase 1, which is where homologous pairs separate, giving two new haploid sex cells in telophase 1, which each have 23 chromosomes and 46 chromatids because they still have their duplicates. Now, this is going to allow cells to go through the second round of cell division, in which later on in anaphase 2, they're going to split up the sister chromatids from each chromosome, giving a total of 4 haploid sex cells in telophase 2, each with 23 chromosomes. This concludes our virtual biology lecture on mutations and meiosis. I hope you all took great notes and wrote down any questions or concerns that you may have so that we can discuss in class. Well, that's it. Have a great one.
Teach me some meiosis, teach me some meiosis, teach me some meiosis, teach me some meiosis.